Welcome to today's lesson on culturing microorganisms. In this session, we will explore the techniques used to grow bacteria and understand the laboratory methods involved in the growth and study of microorganisms. In today's lesson, we will firstly introduce the importance of studying bacteria by culturing them and understand what bacteria need to grow. Secondly, we will understand how to culture bacteria using the nutrient broth solution method. Finally, we will learn about how to culture bacteria using the agar plate method. So far, you have developed an understanding of the basic structure and function of different types of cells, such as prokaryotes, like bacterial cells, and eukaryotes, like plant and animal cells, as well as their role in various ecosystems. This foundational knowledge helps us understand the basic structure of a bacteria cell and appreciate why and how we study bacteria in controlled environments to gain further insights. Let's get started. Today, we will explore how we study bacteria by growing them in the lab, a process known as culturing. Culturing microorganisms is an essential technique in microbiology that allows scientists to study bacterial evolution, genetics, and responses to various conditions. The concept of culturing microorganisms advances our biological understanding by allowing scientists to observe their growth patterns, understand their nutritional needs, and study their interactions with different environmental factors. Before we look at the techniques, it's important to understand what bacteria need to grow. Firstly, bacteria require nutrients. These nutrients are provided in a culture medium and include carbohydrates for energy, proteins, vitamins, and minerals. Secondly, bacteria must grow at a suitable temperature. Most bacteria grow best at temperatures similar to those of the human body at around 37 degrees Celsius, but some may require different conditions. Finally, bacteria sometimes need oxygen to grow. This type of bacteria is called aerobic bacteria. However, some bacteria do not need oxygen to grow, and these are called anaerobic bacteria. There are two primary methods for culturing bacteria in the lab. These are the nutrient broth solution method and the Petri dish, or agar plate method. Let's begin by learning about the nutrient broth solution method. For this method, we must firstly prepare a sterile culture medium, known as nutrient broth. This solution is rich in carbohydrates, proteins, vitamins, and minerals. Next, a suspension of bacteria is mixed into the nutrient broth within a flask. This is known as inoculation, as an infectious material is introduced. To prevent contamination, the flask is stoppered with cotton wool, as this keeps contaminants out. Next, the mixture is shaken regularly to provide oxygen, promoting the growth of aerobic bacteria. This stage is known as aeration. Finally, the bacteria undergoes incubation, during which the flask is placed in an incubator at a suitable temperature to allow bacterial growth. The nutrient broth method is useful for growing bacteria quickly and obtaining a large number of bacterial cells in a liquid medium. The second method, used to cultivate microorganisms, involves using agar plates, a Petri dish containing a growth medium. Let's break down this process. To prepare the medium, agar, a gelatinous substance derived from seaweed, is mixed with nutrients and poured into Petri dishes, where it solidifies to form a gel-like surface. To inoculate the medium, wire loops called inoculating loops are sterilized and dipped into a bacterial solution. They are then streaked across the surface of the agar to spread the bacteria. After inoculation, the Petri dish lid is securely taped to prevent contamination. Finally, the sealed Petri dishes are inserted into an incubator for a few days at a suitable temperature, allowing microbial colonies to grow on the agar surface. The agar plate method is advantageous for isolating and studying bacterial colonies. Individual colonies can be seen and identified, making this method essential for microbiological diagnostics. Before we conclude, let's discuss how one of the most influential antibiotics was discovered accidentally using the methods we have learnt about today. The discovery of antibiotics was a turning point in medical history, and one of the most surprising moments came when Alexander Fleming stumbled upon penicillin in 1928. Fleming, who was known for his somewhat disorganized lab, returned from a holiday to find that a mold had contaminated one of his bacterial cultures. Instead of discarding it, he noticed something extraordinary. Let's take a look at what happened.
I'd like to share with you a moment that changed the course of medical history, a discovery that came about quite by accident in my own laboratory. In 1928, I had just returned from a holiday to find that one of my bacterial cultures had been contaminated by a mold. What I observed was extraordinary. The bacteria surrounding the mold were being destroyed. This led to the discovery of penicillin, the first true antibiotic. It's remarkable to think how this paved the way for a medical revolution, saving countless lives since then.
In today's lesson, we explored how bacteria can be studied by growing them in the lab using nutrient-rich media. We began with an overview of why culturing microorganisms is crucial for scientific research and medical applications, such as discovering new antibiotics and understanding disease mechanisms. We then discussed how bacteria can be grown in a controlled laboratory environment in order to study them, and the conditions that were needed for this growth. We then discussed the different types of nutrient media used for bacterial culture, including broth cultures and agar plates using Petri dishes. Broth cultures are liquid media used for growing large quantities of bacteria, whilst agar plates provide a solid surface for bacteria to grow on, making it easier to isolate individual colonies. Additionally, we reviewed each process, including the incubation process, detailing the optimal temperature and time required for bacterial growth, typically around 37 degrees Celsius for most human pathogens. By understanding these culturing techniques, students can appreciate the critical steps required to study bacteria safely and effectively in a laboratory setting. This foundational knowledge is essential for developing further skills in microbiology and understanding the role of bacteria in health and disease. Watch the rest of the GCSE biology course at Sophos Education.